Okay, we're going to switch real quickly to go through TB, and then I'll switch to the helminths. So TB is a bacteria, still a complex bacteria. It has a very, very large genome. If you remember, we were talking about 30,000 base pairs for SARS-CoV-2, and now we're talking about 4 million base pairs for TB. It has 250 genes in its genome just dedicated to fatty acid metabolism, and that helps it form this very wax-rich cell wall composed of these long-chain fatty acids and glycolipids, which are all going to be really great antigens for our innate immune response to recognize that we've been infected with TB. It is very, very slow. And so when we think about our simple 8-gene influenza, 6-hour replication time, really, really fast, get in, get out, infect the next person, versus TB, which takes this very, very slow replication time, has this incredibly complex metabolism, lots of requirements from the host. This is another example where it's going to be with you for decades because it's invested a lot in working with you to be a successful pathogen versus something like a lot of the viruses we've talked about. This is just a map of where it's endemic worldwide. With TB and malaria, TB is actually more prevalent than malaria, over 1 billion people infected, still a tremendous problem. And this just summarizes some of that. I'll mention this is a useful way of thinking about the impact of infections is this disability adjusted life years. So beyond just mortality, there's also loss of quality of life, 2 billion individuals infected, and after you get infected, many people control using the immune responses that we're going to see shortly. But 10% of those, and when you have 2 billion people infected, that's 200 million people, have a risk of developing disease following infection. And if you don't treat the disease, the mortality is 50%. So 100 million people of the 2 billion people that are infected. And a lot of the other people are actually still quite sick and undergoing a lot of damage. If you've taken the between host modeling and thought about R naught, TB has a very high R naught, so 10 to 15 individuals infected by a single untreated patient. So the life cycle is simpler than, in a way, than anything we've talked about. It's just that TB is very, very, very good at what it does. So it replicates and it accumulates in macrophages, mostly in the lung. And so I had shown you pictures earlier of what a lung airway looks like. You imagine this is a big sphere of an alveolus up here. And you have these macrophages, usually one in each alveolus, that just sits there and lives there and just makes sure everything is okay, cleans up any dust and debris that you eat, will activate an immune response if it thinks it's necessary, if it gets activated from the innate immune system. And TB has chosen to live in these cells. So this is the cell that should actually kill most bacteria that come in. And then TB has said, this is where I'm going to set up my house. Anytime a pathogen does that, where it directly attacks the immune response, you know it's probably a pretty sophisticated pathogen. And so the other really great example of this is HIV, where it goes really right for the CD4 T cell at the heart of your immune system. And so it's a much more formidable foe in the sense that it is really attacking you at your defenses. So it preferentially infects the macrophage. It then tricks the macrophage into thinking, well, it doesn't trick it. The macrophage knows there's an infection and it goes about trying to set up an immune response and migrates out of the sort of harsh airspace and starts moving into the tissue of the lung to start bringing in other cell types. And it brings in other cell types and sets up what's called a granuloma, which is this sort of long-term stable structure that's inflammatory, but that's working to try to control the infection. But in many cases, and we'll talk about the reasons why, TB can't be controlled by the macrophage. So the killing effector mechanisms that the macrophage is trying to use against it are not completely effective. They were completely ineffective. You would die acutely very quickly of the disease, but these are sort of long-term and effective. And so you accumulate these granulomas, which themselves contribute to the damage in your lung. They cause this localized inflammation. They cause fibrosis and scarring, but they can restrain the bacteria for a very, very long time. And then you can get reactivation of the disease, and the bacteria will break out of the granuloma. They'll start growing and growing and growing, kill all of the cells that they're in, and you'll cough and you'll spread this to 10 to 15 people. This is a little bit what these granulomas look like. They're very unique structures. Cells actually fuse together, so you get these giant cells that have multiple nuclei. Again, I think this points to the long evolutionary history we have with this pathogen, where there's sort of unique immune responses that have formed just to deal with it. And this is a pretty effective mechanism. 90% of people live their reproductive years without getting severely diseased from TB. And that's the scale that evolution is working on. It's not the scale that we might like to work on, though, if we want to live a very long, healthy life. This is what a lung looks like, which, you know, shouldn't look like this. It should be sort of one nice, solid pinkish color after years and years of accumulating TB growth. So I mentioned briefly the lots and lots of fodder for the innate immune system, these mycobacterial ligands that will trigger the TLRs, start the inflammatory response, get the whole gang going to try to control the bacteria, but will fail to do so in many cases in an effective manner. So you have this feed-forward inflammation from the infected macrophage calling in lots of other cell types, calling in other macrophages, which are now what can get infected. Again, this is very reminiscent of what HIV does, where it calls in more CD4 T cells, which are just more fodder for the HIV to recruit. But eventually these macrophages will call in CD4 T cells, and with all of these classic type 1 cytokines, TNF, IL-1, IL-6, you'll start setting up the type 1 interferon gamma driven immune response, which is going to try to kill intracellular pathogens just like TB. Why is TB so good at this? So remember, it has all of these genes dedicated to metabolism and to manipulating metabolism and to their own metabolism and the host metabolism. And so they'll go into an endosome, just like a virus, in the cell that they're infecting. And one of the ways, the reason cells do this is they're going to fuse that endosome with a lysosome that's going to be full of toxic chemicals and enzymes that should kill anything that's in there. We talked a little bit about that yesterday. What TB does is it releases all kinds of modulatory metabolic regulators that sort of freeze this process from happening. And really a big determinant of whether or not a particular host is particularly susceptible to TB is how susceptible they are to this freezing process. If their endosomes are really, really good at getting to the lysosome very quickly, then they can control TB. If they're not, then they're going to be very susceptible to TB. There's probably some sort of trade-off here with evolution in terms of, if you're very, very good at this process, you might also be more prone to sort of inflammatory conditions and autoinflammation. And so there's a balance between, you know, chucking everything in your extracellular environment into the lysosome versus being susceptible to TB. So in the granuloma, then you're going to have this infected macrophage that's in conversation with all kinds of immune cells, particularly the professional immune cells like the CD4 T cell, that's telling the macrophage by secreting cytokines and by interacting with cell surface receptors, kill the bacteria, kill the bacteria, kill the bacteria. And again, based on sort of genetic variation among the population, some people are better at that than others. CD8 T cells can also try to kill the whole macrophage. You have a number of other cell types that come in and try to participate in this signal to the infected macrophage to try to control the bacteria. And again, we're talking about this as the type 1 response, and that's going to be in contrast to what we talk about in about 4 minutes about the type 2 response, which is characterized by different cytokines, IL-4, IL-5, IL-13, different transcription factors. These two responses are often seen as very opposed to each other because they're opposed to each other transcriptionally and regulatorily, where the type 1 response is really characterized by interferon gamma. In all types of immune responses, and you're going to see this even in the context of type 2 responses, there's always a negative feedback induced in some way. And so even while you're having this massive feed forward, try to get rid of the bacteria, you're having this signal to dampen the immune response and not cause too much damage to your lungs, because all of this inflammation is causing damage to your lungs. And that's going to be mediated by a special subset of CD4 cells called the FOXP3 positive T regulatory cell. And so this is just another T cell that comes in and says, everybody calm down. It's not that bad. We can live with this. We've lived with this for a few years now. We don't want to destroy the lung over whatever this is. And again, if there's polymorphisms across the human population that determine some of that balance. And yeah, so this slide summarizes that. It's really this balance between the regulatory signals and the activation signals that will drive either the classically activated macrophage to have microbial activity, or to be suppressed by the Treg response, or to be suppressed because it's mounted a Th2 response, which isn't going to really help in the infection. And we're going to talk about why you might mount a Th2 response at the same time you should be dealing with TB in a couple of slides when we get to the helminths. IL-10 is a critical cytokine that you're going to see in a lot of studies about TB, because if you make a lot of IL-10, it helps regulate the pathology, and so it can suppress some of the tissue damage and help you reduce symptoms, but it can also suppress the effector mechanisms. It does suppress the effector mechanisms. And so IL-10 can be a sign of a frustrated inflammatory response that's not working, and so you're turning on IL-10 to sort of shut down some of the damage caused by the inflammation. But it is also, if you don't have IL-10, you'll often have a really severe cytokine storm type phenotypes. So this summarizes everything I just told you very quickly. You'll see here the key cytokine here is interferon gamma, and it's produced by many different cell types, and it's all being fed onto that macrophage to try to keep those bacteria in check and not let them reactivate and burst out of the cell. We know this, again, just like in malaria by analogy, by using animal models, which again, probably a little bit better for TB than they are for malaria, but still have their issues. So if you look at the growth of TB in various different knockout animals, if you knock out class 1, which is going to get rid of all your CD8 T cell activity, it's not a big loss. If you knock out class 2, it's going to get rid of all of your CD4 T cell ability. Now it's starting to look pretty bad. If you knock out all your T cells, it looks a bit worse. If you knock out NOS2, which is one of those enzymes that makes those chemicals that kill the bacteria, this is very bad. This implies that NOS2 is the major effector mechanism in mice, at least, that kill TB. And if you knock out interferon gamma, just like in malaria, it's uncontrolled growth of the parasite. And we know this is actually a pretty good model because this is the same thing that happens in humans. There are humans that are born with deficiencies in interferon gamma signaling. Either they don't have interferon gamma because of a mutation, they don't have an interferon gamma receptor, they don't have IL-12, they don't have something in the interferon gamma pathway, and they can't make an interferon gamma response. And virtually all of those people, the way they first present, despite the fact that interferon gamma is used in all of these different types of immune responses, is with various weird mycobacterial diseases, not necessarily TB. There's a whole big family of mycobacterium that you don't get infected with because interferon gamma is so effective at mounting responses to get your macrophages to kill all of the other mycobacterium except for TB. And so there's a chart of this in the online, but you'll see the things like Mycobacterium bovis, which is the mycobacterium that normally colonizes cows. People will get infected with it if they're deficient in interferon gamma because they don't have this arm of the immune response to keep that under control. And so I've shown you 2 examples here now of 2 intracellular pathogens that are controlled to varying extents by this type 1 response, which is either activating cellular responses to kill infected cells or activating actually the infected cell itself, in this case, the macrophage, to kill the bacteria that are inside the cell.